pancreas is a critical organ that performs both endocrine and exocrine functions. In this video, I will focus on the exocrine functions. The pancreas develops as two separate buds, the ventral and dorsal pancreas. The dorsal pancreas grows rapidly, while the smaller ventral pancreas rotates and eventually fuses with the dorsal pancreas, forming the uncinate process. The pancreatic ducts usually fuse to form the main pancreatic duct. Secretions from the entire pancreas exit through what was originally the ventral duct. However, there are several anatomical variations. Pancreas devisum is found in up to 10% of the population and occurs when the fetal ducts fail to fuse. As a result, the main pancreatic duct is truncated and drains only a small region of the head. The rest of the pancreas is forced to drain through the much narrower minor duct, which increases the risk of chronic pancreatitis. Angular pancreas occurs when pancreatic tissue completely encircles the duodenum, resulting in bowel obstruction. The volume of the pancreas increases until around age 40. After age 60, the pancreas begins to atrophy, and exocrine tissue is replaced by fatty tissue. The pancreas is situated retroperitoneally in the C-curve of the duodenum and extends as far as the hilum of the spleen. The pancreas is 12 to 20 centimeters long and is partitioned roughly into 5 regions: the head, neck, body, tail, and uncinate process. The body region drapes across the lumbar spine and is the region most vulnerable to trauma. The neck is relatively thin and lies directly in front of the portal vein. Spread of cancer to this region can make resection impossible, which contributes to the poor prognosis of pancreatic cancer. The pancreas is enclosed within capsule-like connective tissue and partitioned coarsely into lobules. The pancreatic duct of Versung and the common bile duct merge and empty into the duodenum through the hepatopancreatic ampulla of Acinar. A set of 3 circular muscles called the hepatopancreatic sphincter of Oddi prevents reflux into the pancreatic duct. The accessory pancreatic duct of Santorini is present in about 70% of the population. The minor duct drains the uncinate process and part of the head into the duodenum through the minor duodenal papilla. Blood supply to the pancreas is complex and forms an anastomosing network that helps the pancreas to monitor blood glucose concentrations. Branches of the common hepatic artery supply the head to the gastroduodenal artery, and the uncinate process through the pancreaticoduodenal artery. The superior mesenteric artery also supplies part of the head, while the splenic artery supplies the body and tail. As with other organs of the gastrointestinal tract, blood from the pancreas returns to the heart through portal circulation. However, the pancreas also has a secondary portal system in which venous blood exiting the islets of Langerhans passes directly through exocrine tissue, allowing direct local action of endocrine hormones. Venous blood from the head drains into the superior mesenteric vein, while the body and neck drain into the splenic vein. These veins merge into the portal vein and deliver islet hormones directly to the liver. Lymphatics in the pancreas are also extensive and intricate, further contributing to the poor prognosis of pancreatic cancer. The pancreas is innervated with a complex system of sympathetic fibers from the T6 to T10 thoracic splanchnic nerves, as well as parasympathetic fibers from the posterior vagal trunk. However, the pancreas is primarily regulated by cholecystokinin and secretin secreted by epithelial cells of the duodenum. Well, the word pancreas means all flesh. The pancreas is a complex, heterogeneous organ that contains at least different cell types. The Acinar, ductal, and islet cells all differentiate from a common progenitor lineage. Most of the cell type diversity is found in the 1 to 2% of the pancreas dedicated to endocrine function, which contains alpha, beta, gamma, delta, and epsilon cells. The key exocrine cells are the Acinar cells, which constitute 95% of the mass of the pancreas and produce a mix of extremely potent digestive enzymes, including exopeptidases, endopeptidases, lipases, nucleases, and amylase. The nucleus and regulatory receptors are found at the basal region of these wedge-shaped cells, while the apical region contains inactive enzyme precursors stored in membrane-bound zymogen granules. Cup junctions allow chemical and electrical signaling between Acinar cells. Zymogen granules fuse with the apical membrane and are released into the lumen. At the terminals of pancreatic ducts, centroacinar cells are cuboidal epithelial cells that extend into the lumen. They contain carbonic anhydrase and secrete a bicarbonate-rich fluid when stimulated by secretin. Intraacinar ductules merge into larger ductules lined by mitochondria-rich columnar epithelial cells that secrete mucin via the cystic fibrosis transmembrane conductance regulator, or CFTR. Aberrant expression or mutations in this gene reduce bicarbonate secretion, affecting the viscosity of pancreatic fluid and resulting in obstruction. Given the potency of the digestive enzymes, the pancreas maintains several fail-safe mechanisms to prevent self-digestion. Most of the enzymes require an acidic pH and are inactivated by bicarbonate in the pancreatic fluid. As the pancreas lacks starch and triglycerides, amylase and lipase are secreted in an active form, while trypsin is released in the inactive form trypsinogen. In the duodenum, duodenal enteropeptidase, also called enterokinase, cleaves trypsinogen into trypsin. Activated trypsin then catalyzes activation of chymotrypsinogen, phospholipases, elastases, and other proenzymes. However, trypsin has a self-recognition site and also catalyzes its own inactivation. Ductal and acinar cells also secrete trypsin inhibitors such as SPINK1. Unfortunately, symptoms of pancreatic disorders are nonspecific and can remain undiagnosed unless there is a high degree of suspicion. Acute pancreatitis is characterized by reversible inflammatory lesions that can range from localized fat necrosis to widespread necrosis of the parenchyma. This is due to inappropriate activation of pancreatic enzymes, resulting in autodigestion, particularly involving lipase, since it is already secreted in an active form. On the other hand, inappropriate activation of trypsin is a critical trigger for pancreatitis, since it's capable of activating other proenzymes. Although alcohol abuse and biliary tract disease are the most common causes, in about a third of cases, the etiology is unclear. The cardinal symptom is abdominal pain, which can range from uncomfortable to incapacitating. Lab findings reveal a marked and rapid increase in serum amylase, followed by a slower increase in serum lipase levels. Acute pancreatitis is a medical emergency involving systemic release of digestive enzymes and activation of an intense inflammatory response that can rapidly lead to shock. Ultrasonography findings can include swelling, inflammation, calcification in the presence of pseudocysts, gallstones, or lesions. Treatment involves supporting therapy and total restriction of food and liquids. Most patients recover, but in up to 60% of cases, necrotic tissue becomes infected, and 5% of patients die from shock during the first week. Nevertheless, in principle, damage due to acute pancreatitis is still reversible. In the next video, I will talk about irreversible damage and progression to chronic pancreatitis. While the pancreas is essential for life, a failing pancreas also poses spectacular risks, ranging from shock and self-digestion to diabetic coma and death. As the surgical adage goes, you don't mess around with the pancreas.